Okay, so now we are back to kind of what we started at the beginning. We are going to talk about each of the organelles inside of a eukaryotic cell. So remember, a eukaryotic cell is a cell that contains organelles to begin with, but includes plant cells as well as animal cells. So any animal you can think of, or any plant, these are the cells that we're talking about, and we are going to talk about all of the organelles that make up both of them. Remember, we discussed those at the beginning of the lecture. And the things that are different between them. So we're going to start with the nucleus. The nucleus is the cell's genetic control center, so it contains or houses all of the DNA for the cell, and the DNA never ever leaves the nucleus. It always stays inside of the nuclear envelope and is protected because of that, so it doesn't go out into the rest of the cell where it could be damaged. The nucleus is the largest and most prominent organelle in most eukaryotic cells. So if you actually look at a cell, you can see that it's generally purple in color, and it is very, very large. It is the largest thing inside of the cell, and it's kind of a big ball or a big sphere, oftentimes in the middle of the cell. So very prominent organelle. Because it contains so much stuff. It contains all of the DNA, and again, since it's so important, it takes up a lot of space. It's allowed to move around. So its function, storehouse for hereditary information, so it houses the DNA in the form of chromatin. On the next slide, we're going to talk about chromatin and what makes up chromatin, but DNA is part of or is chromatin. It is the genetic control center. So it controls access to the DNA. So there are actually little pores inside of the nuclear envelope that allow certain things to get into the nucleus and certain things to be able to leave. So it controls which molecules are produced and in what quantity they are produced. So it directs most cellular activities. So it's going to be the place where we house our DNA, how we control all of our genetics and control access to the DNA, and then what's produced and in what quantity they are produced is all inside of the nucleus. So those are the functions of the nucleus. I mentioned that the nucleus contains chromatin, so it's DNA in the form of chromatin. Chromatin is a mass of long, thin fibers consisting of DNA with some proteins attached. We're not going to talk too much about chromatin. There are a lot more specifics, but basically DNA is so incredibly large, such a very, very long strand or several strands of these nucleotides, which we talked about in Chapter 2. And since it's so long, we kind of have to condense it in some way. So even when it's in its most uncondensed form, which is chromatin. This is where it's just kind of hanging out, relaxing inside of the nucleus. The DNA is slightly wound around these proteins. So they're kind of like little spindles to where we can take the DNA and wrap it a couple times around a protein and then take another protein and wrap it around a little bit again to kind of condense it just a little bit, even though in the form of chromatin, it's not quite all the way condensed into what we would normally call a chromosome. And we'll see that when we get to those chapters specifically on mitosis and meiosis. But so chromatin is the slightly condensed form of DNA where we're wrapping the DNA around these individual proteins to just kind of get it into a smaller area because it's so extremely large. Also within the nucleus, we have the nucleolus. So the nucleolus is an area near the center of the nucleus where subunits of the ribosomes are assembled. And so we'll talk specifically about ribosomes, but we've already mentioned that ribosomes are like the little protein factories. This is where we have our RNA or mRNA come together with our amino acids, and they put together and create or make up our proteins. So the ribosomes are really kind of these factories. They just pump out the proteins based on what the RNA says. And remember, the RNA says something based on what the DNA says. So in the nucleolus, this is where we have this area where the ribosomes are made, because the ribosomes are actually made up of RNA. which again comes from DNA or is made from DNA. So the nucleolus is this very condensed area, and it has all of the information for the ribosomes, which are these protein factories. So here's our picture. Here again is our eukaryotic cell. This is an animal cell. You can tell because there's no cell wall around it. Our functions in the nucleus are genetic control center, cellular activity, and hereditary information storage. And you can see they've kind of blown it up here. And here they're showing the chromatin, which are the thin fibers of DNA carrying the hereditary information. So it's slightly condensed, but not completely, because it's taking up all of that area, all this kind of light blue area inside of the nucleus. So it's kind of spread out a little bit still. So you can see they show the nucleus as this sphere, and in this sphere we have three kind of regions, or three things going on. We talked about the nucleolus, so that's this very dense center where the ribosomes are assembled. We have the area that has the chromatin, which is the light blue area carrying all of this hereditary information. And then on the outside we have our nuclear membrane, or nuclear envelope. And we have that blown up right here. So it's kind of this plasma membrane, just like we normally would have, but it has pores in it. So you can see this very continuous nuclear membrane, but then it has pores in it, similar to our gap junctions. These pores are going to allow our RNA to leave the nucleus. Remember, our DNA never leaves the nucleus. It always stays in our nucleus. It's protected. But these pores are going to allow the RNA to be copied from DNA and then leave the nucleus and go into the outside of the cell. So it's important to remember that we have this nuclear membrane that is two bilayers covered in pores that surround the entire nucleus. And so we have this very protected nucleus, but then we still have these little pores that allow things to move out of the nucleus, and those things being RNA. So next we can talk about our cytoskeleton. So remember, the cytoskeleton is kind of, as its name implies, cyto meaning cell, and then skeleton. So it gives shape, or is the skeleton of the cell. So it acts as the inner scaffolding of the cell. It provides shape and support to the cell, and it controls intracellular traffic flow. So kind of the first two are given. An inner scaffolding, meaning it kind of gives this shell to the cell, allows it to maintain its shape. So provides shape and then gives support, just like our skeleton does. It provides support to our body, gives us a little bit of protection. It also allows us to maintain our shape that we have. So same thing as it does in the cell, the cytoskeleton. Controls intracellular traffic flow. Now that's an important one to understand, is that, remember we talked about the vesicles. When we have endocytosis or exocytosis, we create these transport vesicles. And when we create these transport vesicles inside of the cell, we have to get them moving to the outside of the cell, or toward the plasma membrane, so they can be released outside of the cell. The way that we do that is using the cytoskeleton. So when we make the transport vesicle, our cytoskeleton will actually act as kind of a walkway. So our vesicle will kind of attach itself to one of the fibers of our cytoskeleton, and it will walk along those fibers all the way to the edge of the cell. So kind of like our streets. So wherever our vesicle is made, so wherever we leave from our house, then we end up walking along the streets, and then we take a turn and we walk along another street, and then we get to the edge of our city, or the edge of the cell. So the vesicles use the cytoskeleton in that same way, where they're kind of walking along these the bones, or the filaments or fibers that are inside of the cell in order to get outside of the cell. Or the opposite, when we bring something into the cell, we can use that cytoskeleton to walk the vesicles into the cell where they need to be. It also enables movement. So we're able to kind of move our skeleton to move the cell. So you can think of this as kind of just this ability to move our bones around to fit through a smaller space, which generally we're not able to do. But the cytoskeleton allows for that. It also will be a continuation into the flagellum, which we'll look at in just a moment, so enabling movement of the entire cell. So here we are, cilia and flagella. Both of these allow for cellular movement, but they are also both very different. So the way that they are similar, one, is that they allow for cellular movement. Two, that they are made up of individual protein filaments or protein fibers. And so cilia, what's different between them is that cilia are like little tiny hairs all over the surface of a cell. So if we think of one single cell, we can think of the surface of that, for example, in our respiratory system, in our bronchial tree. When we breathe in, all of the little tiny cells that the air that we've breathed in passes have cilia on them, have all of these little tiny hairs all over them. You can see the picture on the bottom left-hand side here shows the orange part are all of these little hairs. It looks like a carpet. All of these little hairs are made up of these protein filaments, and they're called cilia. Cilia work like oars. So you can see in the upper left-hand corner here that they have a power stroke and a recovery stroke. What that means is that if you stick an oar in the water and you just push in one direction, then you kind of pull it out, put it back in, and push in that same direction. That's exactly how cilia work. They're able to push in one direction, and then they kind of go limp to get back to the other side, and then they push again. What this does is it allows the cells to move something across the surface of the cell. And the reason why I mention this as far as the respiratory system is because that is how the mucus in our respiratory system moves out. So if you're ill and you have lots of mucus, that's because you have some sort of bacterial infection or viral infection. And so you have a lot of mucus built up in your lungs, for example, if you're coughing up stuff. So what's happening is when you breathe in stuff, all of the dirt and the dust and other things that you're breathing in get trapped in mucus that sits inside of your lungs. The mucus is sitting inside of your lungs but on top of the cilia. And then these cilia are beating in one direction and moving the mucus out of your body so that you can get rid of or cough out all of that stuff that you've trapped. So all of the dirt and the dust and other bacteria and stuff. So cilia are very helpful in moving things across cells in our body. So with gentle beating, cilia can move fluid past cells. On the other hand, our flagella move the entire cell. And again, we only see flagella in sperm cells in humans. So that's the one single cell that we see a flagellum. So you can see in the picture that a flagellum, singular for flagella, which is plural, we have one flagellum on a single sperm cell, and it actually moves in a whip-like fashion. So it spins around, it rotates, which is different than the cilia. So it rotates, which allows the cell or the head of the cell to move in one direction. You can see that in the upper right-hand corner, that we have the direction of swimming is to where the heads are at the beginning or at the front, and then they're able to move those tails, spin them around very quickly to move the entire cell. So whereas flagella, with whip-like motions, can move the cells themselves, so rather than moving something across the surface. So in humans, cilia do not move cells. They move stuff across cells. In humans, flagella move the individual cells, not things across them. However, when we get to prokaryotic cells, so when we're talking about bacteria and what we see is that they can use either of these things to move the cell. Because if a bacteria cell is completely covered in cilia, or little hairs, and all of these hairs are beating in one direction, it allows that one single cell to move in the opposite direction. So this, however, we do not see in animal cells. So next we are taking a look at mitochondria. So again, mitochondria may have existed as separate single-cell bacteria-like organisms billions of years ago. Remember, this is our endosymbiotic theory that we talked about. They could have been completely separate organisms that joined in with another cell. And we see this because mitochondria have their own DNA. And in fact, the DNA in mitochondria are specifically and only from the mother's DNA. So when we talk about mitosis and meiosis, specifically meiosis, we'll talk about how the formation of a new person is half the father's DNA and half the mother's DNA. And so it really is half of both. However, all of the DNA for the mitochondria comes from the mother. It's not half and half. So this is another reason why the endosymbiotic theory is such a strongly held theory, is because it only comes from the mother, which means that it may have come from one initial organism to begin with. So the functions of mitochondria: one, act as all-purpose energy converters. So basically, that's their function. They take energy, or they take food, for example, and they turn it into usable energy in our bodies or in our cells. And so we can see the structure of the mitochondria here. In the bottom left-hand corner, we can see that there is an outer membrane and an inner membrane. And that is important when we get to the chapter where we talk about cellular respiration. So we see this outer membrane, kind of the kidney shape. And then we have our inner membrane, which is highly folded. So it has a very high surface area, lots of space. And this is where all of our reactions occur inside the mitochondria. So our outer membrane, our inner membrane, and then we have this membrane space, or intermembrane space. So it's in between the inner membrane and the outer membrane. So this will be important when we look at cellular respiration, but it's important right now that you know that mitochondria are termed the powerhouse of the cell. So they are the little tiny machines that make all of the energy that we use as humans to function all of our cellular activities, and therefore all of our human functions: our running, our talking, our walking, breaking down food, our thinking. Anything that requires energy comes from mitochondria as making it from food. So next we are taking a look at lysosomes. Lysosomes are round, membrane-enclosed, acid-filled vesicles that function as garbage disposals. So they are basically floating garbage disposals in the cell. So you can see on the left-hand side, we can see our eukaryotic cell here. You can see the little green spheres, which are blown up right here, and then they cut one in half so you can see the inside. So our lysosomes are actually vesicles that contain digestive enzymes. So we've talked about vesicles. You can picture vesicles made up of a membrane. So round and membrane enclosed, just like a vesicle. But then it's filled with digestive enzymes and acids. The digestive enzymes and the acids are so that they can digest things. So if we have our white blood cell rooting around through the bloodstream and it comes upon a bacteria, we know that we do not want that in the body. And so we go through phagocytosis. Remember, phagocytosis, we recognize that the bacteria is not wanted. So we use our false feet, or pseudopods. We have this invagination of the cell, and we grab onto that bacteria. And we suck it into the cell in a vesicle. Then our lysosomes come along, and they merge with that vesicle that has the bacteria in it. When it does that, it uses those digestive enzymes and the acids to then break down the bacteria that are in, or that's been engulfed, that's in that vesicle. So it uses these digestive enzymes to break down that bacteria, and then it will release all the little parts of it into the cell, the parts that are not toxic, so that we can reuse them. So we'll gobble up this bacteria, reuse the parts that we need, and then dispose of the toxic parts. So we'll release that as waste into the bloodstream. Also, you can see in the picture it says a partially digested organelle. And what that means is that, so it shows here mitochondria, for example. Over time, things get worn out in the body. So, for example, our organelles, like mitochondria, start to not do their job as well. They're not functioning as well. So then they need to be broken down, recycled for parts so that we can make new ones. And so the lysosomes will do that as well. They'll engulf some old or worn out organelles, and then they will break them down or digest them for their parts. So not only stuff that we get from the exterior of the cell, but also any old or worn out organelles inside of the cell. So all of these things are digested. We recycle the products that we need, and then get rid of the toxins. Sorry. Next, we can take a look at our endomembrane system. So here is the overview of our endomembrane system. Our endomembrane system includes the rough endoplasmic reticulum, our Golgi apparatus, and then our smooth endoplasmic reticulum. So when we talked about it at the beginning of this chapter, we mentioned that all three of these things are kind of these membranous sacs that are all squished or flattened. So we can see that again here, that we have our rough endoplasmic reticulum of flattened sacs here that are covered in ribosomes. We have our Golgi apparatus, flattened sacs, and then we have our smooth endoplasmic reticulum, also flattened sacs. And you can see those here. So our endomembrane system includes all three of these organelles. The function is to produce and modify molecules to be exported to other parts of the organism. So using our rough endoplasmic reticulum, our smooth endoplasmic reticulum, and the Golgi apparatus, we make molecules that then need to be exported, or need to be shipped out of the cell and sent to other parts of the body. Also, the function of the endomembrane system is to break down toxic chemicals and cellular byproducts. That's mainly the function of the smooth endoplasmic reticulum. So the rough endoplasmic reticulum and the Golgi apparatus function to create things that need to be shipped out of the cell, and our smooth endoplasmic reticulum is responsible for breaking down toxins and cellular byproducts. So first we'll take a look at our rough endoplasmic reticulum. Again, the function of it is to actually modify proteins that will be shipped elsewhere in the organism. So remember that our rough endoplasmic reticulum is termed rough because it has ribosomes completely covering it. You can see here in the picture the rough ER, endoplasmic reticulum, is covered in ribosomes. So ribosomes are the protein-making machines. So what happens is the ribosome, again, will meet up with our RNA from the nucleus, and it will make a protein. Remember, when we talked about proteins, we talked about our primary structure, the initial just chain of amino acids attached to each other. Then we moved into our secondary and tertiary structures, where we have those amino acid side chains, or R groups, all functioning or all interacting with the other R groups. And when that happens, that actually happens inside of the rough ER. So our ribosomes will make our primary structure, just that string of amino acids, and it pushes that string of amino acids into the rough endoplasmic reticulum. When it pushes that primary structure into the rough endoplasmic reticulum, Then it allows it to bend and fold and turn into those coils and those sheets that are pleated. And then it allows it to kind of bend on itself. And then the rough endoplasmic reticulum will change proteins if necessary. So it can add certain things to it. So for example, if we want a glycoprotein, glyco meaning carbohydrates, and then protein. So the rough endoplasmic reticulum will add the carbohydrate and attach it to that protein to make the glycoprotein. So once the rough endoplasmic reticulum has done this, has modified the protein, then it will gather up a whole bunch of these modified proteins and it will create a vesicle. So it will kind of glob out a vesicle. So you can see if we make it inside of one of these flattened sacs, inside here we're modifying the protein, making a glycoprotein. And then what we can see is that it will kind of just create a vesicle. It will glob out and pinch off a vesicle that's filled with these glycoproteins now. I'm going to skip ahead one slide to the Golgi apparatus to continue our protein, and then we'll come back to the smooth endoplasmic reticulum. Sorry about that. So we create this little vesicle that now has our glycoproteins in it. So our carbohydrate and protein mix. This little tiny vesicle, called a transport vesicle, now is going to merge with the Golgi apparatus. So it moves from the rough endoplasmic reticulum, and then it merges with the Golgi apparatus. Once it does that, the Golgi apparatus is going to process it. So it will recognize what type of glycoprotein it is, maybe add something else to it. And then what it'll do is it will package up the proteins, any lipids, and any other molecules for export and send that or ship it out to the other part of the organism. So what that means is I always liken the Golgi apparatus to the FedEx of the cell. So if you take your package, so say it comes from the rough ER, you take your package full of glycoproteins, your bag full of glycoproteins, you bring it to the FedEx, you bring it to the Golgi apparatus. The Golgi apparatus is going to say, okay, where do you want this to go to? All right, we should probably put this in this size box. We should probably put this bubble paper in there. We'll use this tape to seal it so it doesn't get broken open. So it's packaging up these glycoproteins or whatever it is that comes into the cell, the lipids or proteins. So it will package these things up, and then it will mark it to ship to a different part of the cell so that it can be released from the cell and go to the other part of the body. So the Golgi apparatus is kind of like the FedEx, where it's going to ship stuff out, but before it does that, it packages it, makes it all nice and pretty how it's supposed to be before it gets shipped out. So now we'll back up just a second to the smooth endoplasmic reticulum. So kind of thinking in that same way, our smooth endoplasmic reticulum synthesizes or makes lipids, such as our fatty acids, our phospholipids, and our steroids. So the reason this is part of our endomembrane system is because if it's synthesizing or making lipids, then what it can do is it can make those lipids bud off one of those transport vesicles and then bring it to the Golgi apparatus or the FedEx so it can be shipped out. So if we need to ship out a phospholipid, we need to ship out a steroid or a fatty acid, then we can do that via the Golgi apparatus. So smooth endoplasmic reticulum makes our fatty acids or makes our lipids. Also, however, the smooth endoplasmic reticulum detoxifies molecules. So it will actually break down alcohol, break down drugs, and other waste products. It's smooth because it does not have ribosomes. It has nothing to do with proteins like the rough ER does. It only has to do with fats and detoxification. So if you've heard something like your liver is actually what breaks down alcohol, if you drink a whole bunch of alcohol, your liver is working very, very hard. That's why you can get cirrhosis if you drink too much. The liver cells are the ones that actually break down the alcohol, which means that they have a whole bunch of smooth endoplasmic reticulum inside of the cells. So liver cells are packed with huge amounts of smooth ER because the liver is the one that detoxifies harmful molecules. So specifically, we can think of it as detoxifying alcohol. So lots of smooth ER means lots of alcohol or lots of drugs or any other waste product. All right, so here's another look at the entire endomembrane system. So you can see, we can start number 1. Our transport vesicle buds from the smooth or the rough ER. So when we're talking about the rough ER, again, we're talking about proteins. If we're talking about the smooth ER, we're talking about lipids. So the rough ER releases their proteins through a transport vesicle. The smooth ER releases the lipids from or through through a transport vesicle. Then the transport vesicle fuses with the Golgi apparatus and dumps its contents. You can see the Golgi apparatus, and then you can see this vesicle is merging with this Golgi apparatus, and it'll dump all of those molecules inside of this first sac. Then the Golgi apparatus is going to modify the molecules, add the bubble wrap, add the tape, add the box, as it moves through each of these sacs. You can see number 3 on there. It's moving between each of these sacs, getting all these modifications. Then, at number 4, our modified molecules bud off from the Golgi apparatus as another transport vesicle, and then that vesicle will travel throughout the cell, remember, attaching to the cytoskeleton for movement, fuses with the plasma membrane, and then dumps all those contents outside of the cell into the bloodstream. So here's our endomembrane system, which is extremely important because this is how we create so many lipids and create our proteins and then get them to where they're supposed to be going, along with all of these modifications along the way. So next we're going to take a look at the cell wall. And remember, the cell wall is only found in plant cells. We do not find a cell wall in any animal cells at all. So the cell wall is made primarily from the carbohydrate cellulose. Remember, we talked about that in chapter 2. Cellulose is one of our complex carbohydrates, and it is very strong. So strong, in fact, that we cannot break it down as humans, and it makes up the cell wall. And it surrounds the plasma membrane of a plant, and we've talked about that as well, specifically when we are talking about tonicity in our plant cells. So you can see that our cells have our cell wall, this green wall around them, and you can see the cell wall against these 3 different cells here. So they butt up against each other. And remember, we have these tunnels called the plasmodesmata. And you can see the plasma membrane right in the inside of the cell wall as well. So functions of the cell wall. 1, it provides the cell with structural strength. So remember, we cannot actually wilt our plant cells completely because they have this cell wall. It still gives some structure to the plant. So if we put celery in a glass of salt water, we do not dissolve the celery. We still have the cell walls there protecting the plant, but we do have wilting of the celery. Secondly, it gives the cell increased water resistance. So this has to do with osmosis, just as we were mentioning with tonicity. If we put a whole bunch of water into the cell, so if we put it in a, what type of solution? Hypertonic or hypotonic? If we put it in a hypotonic is the correct answer, solution, then the water will move from the outside, where there are less solutes, to inside the cell to dilute the contents. Remember, when that happens, if it's an animal cell, it will blow up the cell. It will lyse. In a plant cell, it only fills up all the way to the cell wall. So it gives the cell increased water resistance, meaning that it does not allow the cell to burst. Then third, it provides some protection from insects and other animals that might eat plant parts. So since it is so strong, since the cellulose is so tough, it can protect from some small insects, from some very small things like bacteria that will try to destroy the plant but cannot get through that cell wall to the actual cell itself inside the plasma membrane. Our next organelle is also only found in plant cells, and it is the vacuole. So you can see it's this very large tan kind of bubble inside of the cell. So its functions, one, it stores nutrients, so kind of acts as a storage cabinet for nutrients, this big empty space to store stuff for later. It also retains and degrades waste products, so it kind of acts as a lysosome where it can break down stuff for the cell. So it's this big expansive area that stores things and breaks things down. It also accumulates poisonous materials, meaning it will kind of collect all the poisonous stuff that we do not want damaging the other organelles so that it can get rid of it eventually. It also contains pigments that enable plants to attract birds and insects that help the plant reproduce. We won't talk about that too much, maybe a little bit at the end of the semester when we're talking about different organisms. So our vacuole functions in that way. And then it also provides physical support. So it kind of acts as this storage area. So it stores nutrients, stores waste products, stores poisonous materials, all these things we want to keep away from the other organelles, with the exception of nutrients, which we're just going to hold on to until the other organelles need them. It also provides physical support in the fact that it will actually hold on to some water sometimes. It'll hold on to water, or it will release water when it is necessary. Then our third difference between our animal cell and our plant cell are the chloroplasts. So we only find chloroplasts in plant cells. Remember that the chloroplast is part of our endosymbiotic or endosymbiosis theory because chloroplasts resemble photosynthetic bacteria. So as that implies, our chloroplast is actually the site of photosynthesis. So plants, again, are able to take sunlight, sun energy, and convert that into sugar. And then it can use the sugar to build up cellulose, build up starch, and then fuel all the cellular activities. So that is photosynthesis, is that conversion of light energy into chemical energy. And we cannot do that, which is why chloroplasts are only in plant cells. But this idea that this one single organelle can do something so powerful is what lends itself to the endosymbiotic theory again. So they resemble photosynthetic bacteria, which leads us to believe that it may have been its own bacteria at one time that merged with another cell. It also contains circular DNA. So similar to the mitochondria, which have their own DNA, it has its own DNA. And then it has a dual outer membrane. And since it has this double membrane, that makes it even more powerful and allows us to believe that it probably was an outer membrane at some point, rather than like all of the other organelles, with the exception of the nucleus, that has a single membrane. So the chloroplasts are the site of photosynthesis for all plants. So where that conversion occurs and where they're able to make their energy. Excuse me. So here's a nice chart that describes all of the organelles that we just talked about. So you can see the structure. So it says the name of the structure. The nucleus, for example, shows you a picture so you can identify it in a cell, which you should be able to do. Tells you if it's in animals, tells you if it's in plants, and then tells you its basic function. So keep in mind that this chart, or these two charts, are just kind of a review to kind of remind yourself, but you should know each of the functions for the different organelles in much more detail than is said here. So if you can just remember that, for example, the Golgi apparatus does this, or the mitochondrion does this. That will help you to remember all of the different functions that we just talked about, which you should know about. So, for example, the rough ER, it says modifies proteins that will be shipped elsewhere. It's important that you know about ribosomes being on the rough ER and how the ribosome makes the protein into the rough ER that then modifies the proteins to be shipped to the Golgi apparatus. So you should know where it gets shipped to. That's part of the endomembrane system, which you also need to know about. So this chart is kind of just a nice rough general outline for you to kind of study from and refresh your memory. So that's it for this lecture. You want to make sure that you know all of these things that we talked about. I know it's a very, very big chapter, talking about lots of different things. Make sure that you listen to all different parts of the narration, and remember to write down any questions you have so that we can discuss them in class. So have a great rest of the day.